Well, hello there. We're continuing our studies of anatomy and physiology of digestive tract, and this is the third video that we're creating. This time, we're going to limit the length of this video to really only address the stomach with its anatomy that was also covered in the very last few minutes of the previous section. And then we're going to spend a little bit more time talking about histology and physiology, explaining different events that lead to proper digestion while food lodges in the stomach and becomes converted into chyme. So let's go to slideshow that we need to share, and let's make this our starting position for today. So we need to just start the slideshow from the current slide. It's going to be this one, and here we go. Basically, the main topics of today's presentation are going to be multiple layers of the stomach wall to find out where they really differ from the rest of the digestive tube. Not only that we're expecting the third layer of a muscle, but also to add more information about the complexity of the mucous membrane itself and a variety of different cells that are displayed within it. Regulation of gastric secretion is going to obviously be how that works, part of presentation, as well as regulation of gastric motility. We know what this leads to. Basically, with a gastric secretion, addition of enzymes, addition of acid, we're pretty much starting the process of chemical food breakdown that will result in absorption of these smaller building blocks within the small intestine. And in order to get to that point, basically the stomach has to gradually release its content into first part of the small intestine, into duodenum, which will allow additional mixture of pancreatic juices, bile, coming from liver and gallbladder in order to make the chyme better suitable for activation of different enzymes that do not like so much acidic environment, but rather prefer something that comes closer to neutral, if not even alkaline. Let's quickly recap the information that you and I already know, so it will be fairly short. There are four basic parts of the stomach that we can identify. They are named the cardia, practically the entry point of the bolus into the stomach. Then the fundus, which we also talked about, naming it and identifying it as the gas trap. Then main part of the stomach, which is the body, of course, and the very last part, which is known as the pylorus. We know that there is pyloric antrum, pyloric canal, pyloric sphincter, but this has been already mentioned in the previous recording. Two additional landmarks worth adding to this review are the lesser curvature, attachment of the lesser omentum, connecting lesser curvature of the stomach to liver, and lesser omentum also would contain very important vena porta, main hepatic artery, as well as biliary ducts. Greater curvature of the stomach, attachment point of the greater omentum, and if you remember a presentation of peritoneum, basically behind the stomach, we're going to have a bit of a large secluded space created by mesentery, and that is going to be called omental bursa or omental sac. Entrance into omental bursa or omental sac is further to the right from the lesser omentum. There would be an opening which essentially allows access into that compartment. Inside, mucous membrane of the stomach is thrown into pretty much intensely formed folds that are named rugae, and they are mostly visible when the stomach is empty. And also, as we know, the third obliquely running layer of muscle is added to the walls of the stomach. It still remains mucosa, submucosa, muscularis, and serosa. However, we're seeing a little bit of a difference. And perhaps the most significant differences are definitely coming from the mucous membrane itself. As one can see on this hand-drawn diagram, which really nicely and pretty accurately depicts the situation, we have mucous membrane, which by itself is not something that you say, okay, these are rugged. No, they are not. The mucous membrane, the fine mucous membrane that you can see here is enlarged under a microscope, is thrown into much smaller, but also quite intensely formed folds that are generating these deep crevasse spaces that are named as gastric pits. Rugae are going to be something that you can see on the far left side of the screen, but this is just like illustration what microscope will identify. Now we can see that it looks like a simple single columnar layer of the epithelium. But if we come close enough, you will find out even in this diagram that not all cells, although they look very much similar, are colored the same. Obviously, that's going to be another task that you and I have to fulfill to differentiate between multiple different types of cells that are forming the mucous membrane. There are also some lymphatic nodules, mucosa-associated lymphatic tissue, loose connective tissue, some blood vessels that are coming in, arterioles, venules, and there should be also some glands that we can see scattered through the mucosa. There isn't much new to add. To submucosa muscularis, we already know the third obliquely running layer of muscle, and finally outermost layer serosa, or in this case we can call it peritoneum because the stomach, as we know, is intraperitoneal structure. Now let's go to the next picture, which will bring one gastric pit with a variety of different cells for us to understand a little bit better as what's going on. You will see on the diagram, and this is what I'm encouraging you to do, to first take a look and see the difference in colors of these cells. And there is one color which dominates, obviously, that is the pink shade, and the first cell on the top of the screen, which says these are surface mucous cells. Obviously, you can get through their name right away idea of what they secrete. It's a very thick layer of mucus that they produce up to 3-4 millimeters thick, which by itself does have a huge capacity to protect mucous membrane of the stomach against self-digestion. Because when the stomach releases the acid, it creates environment which is quite acidic. pH could go as low as pH value of 2, which we know would be acid strong enough to create massive burns of any tissue, stomach included. So with the mucous membrane being formed on the surface of a gastric pit, it practically keeps the stomach safe. And should we go just a little bit deeper into gastric pit, you will also find multiple cells of this color. They're simply known as the mucous neck cell compared to mucous surface cells. They do the same thing except that they fill lower and deeper parts of the gastric pit with additional mucus. Okay, so these two types of cells are pretty much self-explanatory. There isn't much to worry about what they do. But now let's focus a little closer on parietal, chief, and so-called G cells. Parietal cell is really important because it is secreting two different compounds of enormous value. One is intrinsic factor. Without intrinsic factor, we will not be able to properly absorb B12 vitamin. So lack of these cells basically could lead to avitaminosis B12. Secondly, and for what we're studying right now, I think that the role of secreting hydrochloric acid should really go to the very top of what parietal cells are doing. Secretion of hydrochloric acid, without which the digestive process will be fully halted. We need acidity to tenderize the food which came into stomach. We need acidic environment to activate number of different enzymes and eventually to inactivate some of the enzymes that have been already secreted. For example, there would be salivary amylase, which we indicated starts breaking larger molecules of carbohydrates. It will become inactivated when saliva with its enzymes comes into stomach. However, we don't really worry so much about it because additional enzymes that will come from pancreas will continue breaking down the carbohydrates, so the process will not stop. It just inactivates first enzyme that we added on the list. Also, there would be activation of lingual lipase. Well, while released from the surface of a tongue, lingual lipase cannot do anything to start breaking down lipid molecules, but its activation within the stomach actually starts doing the work and later will be complemented with additional enzymes that will complete it. So, parietal cells, hydrochloric acid, and intrinsic factor. Now, we're going to cells that are called the chief cells. The chief cells are secreting pepsinogen and gastric lipase. And I'm sure that you're seeing here right away something that deserves few words of clarification. Lipase, I'm sure that you recognize it with ending ASE as an enzyme. Amylase, lingual lipase, gastric lipase, obviously enzyme. But there could be a little bit of a secret, which is still wrapping up the word pepsinogen. What pepsinogen might be? It's actually inactive form of an enzyme that is called pepsin. And the pepsin will be used to break protein molecules. However, when a chief cell secretes it, it is released from the apical part of the cell in its inactive form, pepsinogen, which will be activated in presence of either semi-digested proteins or in the presence of acidic environment. This is very important, the concept of activation of the enzymes, because otherwise, if the chief cell will have just a production of pepsin within the cell, basically it will be active enzyme which will start breaking down the protein components of the cell which made it. So inactive form to be released and then activated upon its entry into lumen of the stomach. Finally and lastly, we have G cells, and they appear to be just sporadically found on this entire diagram, turquoise blue colored. G cells are hormonal cells that is part of the enteroendocrine function that we talked about in the previous two recordings. G cell secretes the hormone gastrin. So I think it's also easy to remember, G cell, hormone that also starts with the letter G, gastrin. So you see now the variety of different cells, and I'm sure that you are understanding it right away a little bit better as what is going on and what type of cells we have in order to fulfill a variety of different functions that the stomach has to fulfill. Let's go to the next slide, which is just a microscopic photograph with enlargement of about 250 times, where you can see one gastric pit. You can see the epithelial cells. You really cannot say that you have much of a chance to differentiate between them. Most of the cells that you see are surface mucous cells. They appear to be all the same, but deeper in the gastric pit, other cells will be residing, but of course on the photograph it is not so easy to recognize them as it would be on a previous slide on the handmade diagram. So we're going to stop here for a moment on this slideshow, and although it might look quite stationary for a little while, I just want to introduce something for you to understand how the stomach really works. In the first slide, we indicated that we will be talking about regulation of gastric secretion and regulation of gastric motility. Now we already have the knowledge ahead of us which is we understand which cells secrete what. And of course, gastric motility is perhaps something that we can very loosely think about. Okay, well, continuation of peristaltic wave, periodic relaxation of pyloric sphincter, and pretty much 10 to 15 milliliters of chyme coming out of stomach and entering the duodenum. And in essence, that is pretty much it. So, secretion of gastric juice is not done spontaneously. It is controlled by both nervous system as well as hormones. Stimuli in the brain basically will come and stimulate the stomach for increased gastric juice production. We mentioned it before that just seeing the food or smelling the food sometimes could be a stimulus, which is good enough to initiate secretion of the stomach and to start preparing it for eventual arrival of the food a couple of minutes after that into the stomach. Gastric secretion, for that reason, cannot be just identified as, we need to see or smell or taste the food and that's it. Well, it would be just to start. That's why gastric secretion, when we're talking about it, we're dividing it into three different stages, being called cephalic, gastric, and intestinal phases. Once they begin, basically these three different phases could easily overlap each other and practically top up each other's function. And it is what involves both hormonal as well as neural control. So let's talk about these three phases one by one. Cephalic phase is the one, obviously, to start the process. Seeing the food, smelling the food, tasting the food, sampling the food would be already stimulus strong enough. That'll be carried by taste receptors inside the oral cavity into medulla oblongata, where the signal will actually be passed on to vagus nerve, which is to stimulate stomach and its submucosal plexus to start secretion of variety of different things that stomach produces. One is mucus to be increased, the other one is hydrochloric acid, then also production and secretion of pepsinogen. Everything has to go into lumen of the stomach. Also, hormone gastrin, if you remember G cells, is coming out of the cells, but it doesn't go directly into lumen. It actually goes into the bloodstream. Now we can think about second phase, which is the gastric phase, and food, this time termed bolus, enters the stomach. Well, it's not single bite, it will be likely more than that. A little bit of liquid, a little bit of solid, a little bit of saliva, everything mixed up together, getting into the stomach. And as the food lodges in, inevitably it is going to start stretching the walls of the stomach. Sensory receptors within the wall will immediately start detecting this kind of physical stretch. And also there will be other form of receptors that will start immediately indicating change in a pH value. Basically, food we consume is, let's say, neutral. If a stomach has prepared some of its acidic secretion, bringing neutral food in is going to start bringing pH value further up closer to neutral, which means stomach will have to be stimulated to continue with the production of hydrochloric acid in order to meet the demands of regular digestive system processes. Although we say stomach will churn the food as it has the third obliquely running layer, basically we can also think about it as somewhat more stationary form of mixing waves that are happening inside the stomach before relaxation of pyloric sphincter allows for these infamous 10 to 15 milliliters of chyme to enter the duodenum. Once we start releasing chyme into duodenum, we can expect the negative feedback loop which will start suppressing additional acidic secretion because the pH doesn't have to go infinitely to lower and lower value. As the stomach becomes empty, basically its walls start shrinking back to neutral, and sufficient levels of acidity have already been reached. There is no need to further increase it, and that's why the hydrochloric production slows down. Hormonal control is also interesting and important in the gastric phase. Partially digested proteins under, of course, influence of enzyme pepsin, they start also raising the pH value. They act as buffers. And of course, there will be parasympathetic fibers that are releasing acetylcholine that stimulates G cells to release a gastrin. We mentioned that this hormone enters the bloodstream, and then through the bloodstream, it reaches gastric glands that are its target cells. Gastrin stimulates production of gastric juices, and in addition to that, it also makes sure that the inferior esophageal sphincter practically is tightening its own grip to prevent any accidental or involuntary regurgitation of acidic content of the stomach backwards into esophagus. But at the same time, as one sphincter's tone is increased, inferior esophageal, then the pyloric sphincter is gradually receiving the same signals, but the opposite direction, to relax. Stimulation of gastrin or further release of gastrin is going to stop when a pH reaches about level of pH 2 and when the stomach becomes less distended. There would be also additional secretion of histamine, something that we necessarily do not align with the function of stomach. Rather, we're thinking about histamine perhaps along some kind of allergic reactions, but with a histamine role, basically, it is enhancing effects of gastrin on the stomach mucosa. And after cephalic and gastric phase, basically, we're now coming into third part, which is intestinal phase that is controlling gastric secretion. Now we have to start thinking about some kind of reversal. And I'm sure that you would agree with me. Reversal is needed because acidic content comes into small intestine. If it becomes increasingly acidic, basically we're putting small intestine at risk because it doesn't have the same mucous membrane to protect itself against the abrasive acidity that could easily and quickly burn a pretty long segment of the small intestine. So this is going to slow things down a bit. As the chyme enters the small intestine, intestinal reflexes become inhibiting gastric phase of digestion. It will slow down stomach emptying, and the main concern would be not to cause copious amount of chyme coming at once from the stomach. Small intestine would be unable to process that significant increase in volume. For that reason, 10 to 15 milliliters per minute is what we consider optimum. Chyme, which enters the duodenum, is still full with carbohydrates, with fatty acids, proteins, because the stomach did not break down everything. And of course, that kind of interesting chemical composition of chyme will also influence the small intestine to do its own enteroendocrine secretion. The small intestine secretes two hormones, secretin, interesting secretion of secretin, and cholecystokinin. Cholecyst is actually older name for gallbladder. Cholium is bile, cyst is the bladder. So cholecystokinin, obviously some kind of activation. So that is a hormone. Secreting itself is going to slow down until it stops gastric secretion, because there is no further need for increase of acidity, while cholecystokinin inhibits stomach emptying. preventing copious amount of chyme to come at once. Both secretin and cholecystokinin are having direct positive stimulatory effect on the liver, on the gallbladder, and on the pancreas. So Mother Nature obviously did not waste its resources with the same set of hormones. Basically, you're slowing down one part of digestive system while at the same time you stimulate some other parts to increase their activity and to continue with the process of digestion. Mucosa of the stomach, being exposed to really highly acidic content, is, as we mentioned, protected by its own mucous cells, whether they're deep or superficial. Gastric enzymes that can digest proteins are also capable of digesting the stomach itself. So I don't have enough words to highlight the importance of this kind of thick mucosal coat, which protects the stomach. And also, we are not just thinking about the negative effect of acid directly on the mucous membrane. We're thinking about whether acid could eventually find its way between two adherent cells and maybe start affecting deeper layers of mucosa or submucosa. Well, very tight junctions are made between epithelial cells of the stomach, so acid, even if it penetrates through the thick coat of mucus, cannot go much deeper. Secondarily, these cells that are lining up the stomach are also very, very quickly replenished. And we're talking about possibly 4 or 5 days interval, which is enough for renewal of a stomach mucosa, replacement of older, worn out cells with those ones that are new and that will keep doing the work as good as the older one cells that are eliminated would do. Okay, so I'm sorry for keeping it this long, but it really doesn't have any kind of particular diagram that we can use in order to deliver that much of information. So back to what you see right now on the screen. And as illustrated, you're looking at a single parietal cell, and we're trying to figure out how is it possible that parietal cells actually make hydrochloric acid. On the left side is a chyme in the stomach. On the right side in blue, that is submucosa, lamina propria, and some kind of blood vessels. This is the apical end of the cell. This is the basal part of the cell. And let's find out as what's going on. We have to start from within the cell. And this is the process that pretty much you keep hearing about ever since you started studying human body, that end products of our metabolism being water and carbon dioxide under the influence of carbonic anhydrase enzyme are going to form very weak acid, which is carbonic acid, H2CO3. But it's still an acid which is capable of dissociating, and as a result of its dissociation, it yields a single hydrogen ion and bicarbonate ion with a single negative charge. So this reaction pretty much will come again in a urinary system where kidneys are also expected to get rid of surplus of hydrogen ions, and we'll find out how they do it. But in the meantime, we're more focused on to see what's happening with the hydrogen ion here. Okay, looks like we're going to have to follow its trail. And we're talking about some form of active mechanism which has to push content against their concentration gradient. That's why we need adenosine triphosphate as the energy. And what we have on the apical end of a cell is potassium, hydrogen potassium pump, which really allows hydrogen to be exported out of the cell and to allow potassium to be re-imported into the cell. However, take a look. We have a surplus of potassium, but we know that we cannot overload the cell with it. So it does have its own free channels. So potassium, as it comes in, immediately comes out, becoming ready for yet another cycle and another one and another one. So it's almost like some kind of perpetual mobile. On the other hand, we know that chlorine is something that we're also bringing into the cell. So there is a bit of more chlorine ions concentration in the extracellular fluid. So we're using antiport. As we know, this is the protein channel, which allows two different substances to be imported, but they have to travel in opposite direction. Obviously, we're going to get rid of too much of bicarbonate ion by allowing importation of a chlorine ion into the parietal cell. However, chlorine, as it comes in, builds a little bit of extra reserve of chlorine in the cell, which cell by itself doesn't need. So following its concentration gradient, which is higher within the cell than it is in a lumen of the stomach, basically chlorine ions simply diffuse through the apical part of the cell where they meet hydrogen ions, so to say, and of course the formation of hydrochloric acid is something that we have explained through this diagram. We just have to think about what's happening with the bicarbonate ion, basically during this stage of digestion where our stomach produces more significant volume of hydrochloric acid. You certainly expect to have more bicarbonate ion to be produced, released from parietal cells through the antiport mechanism, and then it is going to enter into bloodstream, practically resulting in a somewhat increase in blood alkalinity during this stage, and this is what we call the alkaline tide. I hope that this actually makes sense and that you understand how cells, parietal cells, produce hydrochloric acid, without which, of course, digestion that is happening in the stomach will face a complete standstill. On this diagram, we can now sum up what's happening in three different phases that control gastric secretion. It is cephalic, gastric, and intestinal phase. I intentionally didn't want to use this diagram, but rather allowed you to keep looking at the same diagram and as I was presenting it. So please use the previous couple of minutes of recording where more details about gastric, intestinal, or cephalic phase was presented to you. So if we're to make this as a short recap, perhaps that's what this diagram is really good for. Receptors of a different kind, whether it's eyes or nose or our mouth and taste buds, identifying something as a food or just thinking about food, which is interesting, will start producing signals by cerebral cortex, the uppermost part of it, the most developed, and the hypothalamus that will send signals down to medulla oblongata, which is the brainstem, and through the vagus nerve, basically submucosal plexus becomes stimulated for increased secretion of gastric juice. It's not only hydrochloric acid, you remember, it's also more mucus, it's more pepsinogen, basically more gastrin. And then, of course, as the food enters the stomach, we are looking for immediate addition of gastric phase of digestion and gastric phase of stomach secretion. Stretch receptors detect right away change in the physical shape and size of the stomach, and then the chemoreceptors immediately detect change in the pH value of the gastric juice. I mentioned that food is neutral and it starts rising the value of pH from, let's say, preferred 2 or 3. Basically, it goes up to 4, 5, 6 as more and more food arrives into the stomach. So with the gastric phase, of course, it is going to create more stimuli for additional increase in gastric juice production. So with the activation of pepsinogen, with activation of gastric lipase, of course, the process of chemical breakdown will start. And as we know, there would be a moment when chyme finally starts emerging from the stomach through pylorus and gastric emptying starts. Immediately, we're adding the intestinal phase. Stretch receptors are present in the duodenum. They would immediately detect its own stretch. And the chemoreceptors are going to be there as well, saying like increasingly acidic content keeps coming out from the stomach. So as a result of intestinal phase, enteroendocrine cells are going to start releasing cholecystokinin, CCK, and secreting. Basically, if you follow the red dotted line, you realize it's creating inhibitory hormone effects on the stomach to decrease the gastric juice secretion. And also, we know that there will be increased contraction of the inferior esophageal sphincter, followed and accompanied by relaxation of pyloric sphincter. Enterogastric reflexes via medulla oblongata are also having inhibitory reflex, practically telling stomach your content is now getting smaller and smaller. You have reached and attained for a little while appropriate level of pH value. So time now to start slowing down because this environment actually has done its share of work and we need to move on. I like this diagram, which really nicely puts into perspective as what is happening. Food which enters the stomach disrupts the homeostasis. And in this case, we can say homeostasis was that the stomach was empty for the last 8, 9, 10 hours. Overnight, we don't eat. We don't necessarily eat upon waking up. And of course, the stomach has shrunk to its smallest size. But as we know that food's coming in, basically it is going to start triggering the signals to stimulate secretion of stomach. However, with food coming in, it will immediately have a negative effect on the pH value. It will start going up. Stomach walls will be stretched out. And as we know, chemoreceptors and stretch receptors will detect the change and will create moment to send a signal to control center, which is, again, if we're thinking about cephalic phase, that's fine, which could be stimulation through vagus nerve. And the output, whatever it is, basically is stimulating parietal cells for more hydrochloric acid secretion. Smooth muscles in the stomach wall start contracting more vigorously, allowing mixing up of the content which arrived into stomach with the stomach's own juices. And as a result of that, basically, we're going to have a bit of increase in acidity of the stomach chyme, mixing of the stomach content, and ultimately emptying of the stomach. Now, we realize that this is also a negative feedback loop, like many of other regulatory mechanisms in the body, because as the stomach becomes empty, there is no further need for stimulation of the stomach. So we're following this upward running part of the diagram of a flowchart. Return to homeostasis when the response brings pH of gastric juice and distension of the stomach wall back to normal. That would be like fasting or before we ate. And basically we reach the steady value of pH of the stomach, not being affected by any kind of incoming food. I think that we can also just take a look at this diagram to graphically illustrate stimulation of gastric emptying and inhibition of gastric emptying at the same time, because we know motility of the stomach actually churned chyme and allowed for chyme to be gradually released from the stomach. So if we take a look at what stimulates the gastric emptying, distension of the stomach, presence of partially digested content, whether it's protein, but also like other substances like alcohol or caffeine-containing beverages are going to have same strong stimulatory effect, increased generation of parasympathetic signals in vagus, increased secretion of gastrin. So with gastrin, you will remember contraction of lower esophageal sphincter. It prevents regurgitation of acidic content from the stomach backwards into esophagus, increases stomach motility, so it allows for mixing waves, and at the same time, it is creating a bit of relaxation at the pyloric sphincter, so stomach starts emptying, and this is what stimulates it. However, immediately after stomach, we are entering the duodenum, and the duodenum has the same receptors, detecting stretch, detecting change of its pH, and also detecting presence of some partly digested molecules like fatty acids, molecules of glucose, or partially digested proteins in the duodenum. All of them now are detectable. And as a result of that, basically, you're now seeing increased secretion of cholecystokinin and increased or initiation and then gradual increase of enterogastric reflexes. Basically, if the cholecystokinin is the one that we can say we have its name, the consequence of that is going to be decreased stomach motility, and it will result in some, relatively speaking, inhibition of gastric emptying. In the background, you always have to remember it's the same thing that happens. We cannot allow the stomach to immediately put into duodenum significant volume of chyme, because pancreatic hepatic secretion will not be able to cope with this right away. There would be some kind of pH disbalance, eventually resulting in not proper activation of enzymes, but also it would prevent the entire small intestine to spread the chyme, which becomes 10 to 15 milliliters per minute into small intestine, to spread it out in the form of a thin film, and that will enable small intestine to practically pick and choose and absorb every single absorbable molecule. That is the whole purpose of this kind of exercise. So at this point, I believe that we're reaching the stage where this would be quite good length of a video talking about stomach and some of its physiology. So I suggest that we stop here and practically just mark this as part number 3. Part 4 will take care of the remaining parts that we have need to introduce their anatomy and physiology. Namely, we need to do small and large intestine, and perhaps that's going to be a video on its own before we make one more that will add pancreas and liver and gallbladder into perspective. So I believe that 2 more videos will have to be done. If one, it will be quite long, and I think that everybody's preference is to have something that's a little bit easier, a little bit shorter. And as I say, I hope you enjoy this recording and that you have learned something new. Stay with me. We're continuing this work. New sections are coming soon. Until then, you have a great day. Enjoy it. And thank you very much for watching this video. Bye-bye.